for the opportunity to share my work titled Computational Approach for Drug Repurposing Towards Huntington's Disease. So Huntington's disease is a monogenic neurodegenerative disorder caused by an expansion in the CAG repeat tract in exon 1 of the Huntington gene. In normal healthy individuals, there is less than 36 repeat units, and in patients with more than 40 repeat units, HD is fully penetrant. HD patients suffer from a broad range of symptoms such as impairment to their cognitive, emotional, and motor abilities. And although the disease-causing mutation is known, the mechanism behind Huntington's disease is not fully understood. For example, a prominent feature of Huntington's disease is the inverse relationship between the CAG repeat length and the age of symptom onset, where the longer the CAG repeat expansion, the earlier the symptom onset. However, each CAG repeat length presents its own distinct neuropathology and clinical manifestations. As such, it is unclear how the disease phenotype actually depends on the CAG length. Due to the poor understanding of the disease mechanism, there are currently no effective treatment for Huntington's disease. And as such, my research aims to use computational methods to identify repurposable drugs for Huntington's disease. The idea behind drug repurposing is to identify a new therapeutic use for an existing drugs. And the advantage of drug repurposing is that the pharmacokinetics, the toxicology of the candidate drugs are already established, and this accelerates the process of drug development and reduces cost factors. So the concept of in silico drug repurposing can be illustrated using a figure adapted from Lam et al. So first, we compare the transcriptional abundance between healthy and diseased samples, and this gives us a list of up and down regulated genes known as the disease signature. Second, I use drug signatures from database like the connectivity map, or CMAP in short, which tells me what are the up and down regulated genes caused by the treatment of a drug. And if the upregulated genes in the disease are also upregulated in the drug, and vice versa, we say that the drug phenocopies the disease. Conversely, if the upregulated genes by the disease are downregulated in the drug, and vice versa, we say that the drug reverses the disease signature. So we can then quantify the gene correlation with either a positive or a negative score, where the positive score indicates that the drug phenocopies the disease, while a negative score indicates that the drug reverses the disease signature. And finally, we can then use these scores to prioritize which are the drugs that we can use to potentially reverse the disease signature. So while this concept may seem theoretical, there are actually a couple of drug repurposing successes in the past, for example, in gastric cancer, metastatic colorectal cancer, and epilepsy. So here, my research question is, can I use this approach and apply it into Huntington's disease? So to do so, I took the transcriptional profile of striatal neurons and compared it against the drug database. So striatal neurons, because these are clinically the most vulnerable cell types in Huntington's disease. And what I found is that when I queried this connectivity map database, I found that the drug topotecan consistently and significantly reverses the Huntington's disease profile. So this plot over here shows this plot on the right shows the connectivity score against the ranks of all the drugs in connectivity map, where each gray dot represents a particular drug in connectivity map, and the red circles represent the scores and the rank of the drug topotecan. So from this, one might think that, yes, in silico predicts that topotecan is effective in Huntington's disease, but does it actually work in vivo? So well, in 2016, Sheka et al. Actually treated topotecan onto Huntington's disease mice and showed that it was able to improve HD phenotype, such as the survival rates and motor disabilities of these mice. So this is an elegant proof of concept of the use of in silico drug repurposing in Huntington's disease. And of course, this is not to say that we can leave this room and begin to treat Huntington's disease patients with topotecan, because there's still a lot of work to do. For example, we have to look at the mechanism of action of this drug and how it actually improves the HD phenotype. And this also can give us a clearer idea of what's going on in Huntington's disease. And another thing we can do is to look at the transcriptional profile of other cell types in Huntington's disease, because HD is a brain disorder and it likely affects other cell types in the brain. So it's interesting to explore what are the other drugs that can be helpful for Huntington's disease in the other brain cell types. Thank you.